Today, we're going to look at how cells live their lives, and in particular, how they duplicate to form new cells. I want to start by looking at what it means to have a life cycle in the first place. Let's take the example of humans. We started out as babies and grow up through children until we're adults. As adults, we produce eggs and sperm, and these fertilize each other to make another baby. Cells go through a similar sort of cycle. A newly formed cell grows, prepares to divide, and then finally divides. One major difference between this and the human life cycle is that cells are dividing to make identical cells. In humans, when we have a baby, that baby is not identical to either of its parents. However, cells replicate in such a way that the daughter cells are exact genetic copies of the mother cell. At the beginning of its life cycle, a cell goes through G1, or the first gap phase. During this time, a cell grows and carries out all the normal functions for that cell type. At this point, many cells actually exit the cell cycle and go into what's called a G0 phase. These cells are not destined to reproduce anytime soon. We'll talk about that a little later. If a cell is going to reproduce, it advances into S phase, where DNA is replicated or synthesized. Once a copy of the DNA has been made, the cell enters the G2 phase, or the second gap phase, where it again grows and readies itself for the actual cell division. Cell division occurs through two stages. The first is called mitosis and involves dividing the chromosomes equally into two equal nuclei. Once the nucleus has divided into two, then cytokinesis divides the cytoplasm. As you can see from the outer ring in this diagram, all G1, S, and G2 phases are collectively called interphase. The rest of the cell cycle is the mitotic, or M phase, and that includes both mitosis and cytokinesis. At the end of the M phase, there are now two genetically identical cells. This last point is perhaps the most important thing to learn about mitosis. Mitosis always starts with one cell and ends with two identical cells. Along with interphase, mitosis is like a cellular photocopier. Think back to a long time ago when you were just a fertilized egg. Okay, maybe you can't remember that far back, but at one stage you were a single fertilized egg. You've become big, not by your cells becoming bigger, but by your cells dividing many, many times. Except for your gametes, your eggs or sperm, all the cells in your body have come about through mitotic division. And so it's really very important that DNA synthesis and mitosis happen very accurately so that errors don't accumulate over time. You're probably used to thinking of chromosomes looking like these green ones up here. It's true that they can look like this, but how does DNA get into those structures? Well, in order to package more than 2 meters of DNA into the nucleus of a single cell, the packaging has to be very, very good and orderly. The DNA double helix is wound around proteins into nucleosomes, and these are packaged together into larger and larger structures until we have the shape of a chromosome. Notice that whenever you see this X-like structure, it's actually a replicated chromosome. Both halves of the chromosome are the same, and each side is called a chromatid. Chromosomes consist of DNA and the many proteins around which the DNA is packaged. We'll look at this in more detail in a later presentation. This is what's known as a karyotype, or a picture of all the chromosomes present in one cell nucleus. This picture is taken after cells have synthesized new DNA. That's before the cell nucleus has divided in mitosis. This is a human karyotype and has 23 pairs of chromosomes. Let's look at that in more detail. Here, I'm showing you just 2 of those pairs of chromosomes. Since these chromosomes have already duplicated during S phase, they show the presence of sister chromatids. Sister chromatids are identical copies of one another. They arise through DNA duplication that happens during S phase of the cell cycle. Because sister chromatids are formed by duplication, we expect them to be exact copies of one another. You can also see here pairs of homologous chromosomes. Homologs are similar, but not identical chromosomes. Homologs have the same genes, usually located in the same place along the chromosome, but the DNA sequence is not identical. The reason the DNA sequence is not identical is that one copy is coming from the mother and one copy is coming from the father. Humans, along with most other sexually reproducing species, are diploid. This means that each cell in an individual has two copies of each chromosome. Again, these copies come one from the mother and one from the father. As a result of having two copies of each chromosome, we also have two copies of each gene. We'll see later how this affects our appearance and genetic makeup. Here's another picture of a duplicated chromosome. You can see the two sister chromatids that result from DNA synthesis. Another important feature of chromosomes is the centromere. This is a region of the chromosome with repetitive DNA and a special set of proteins that bind the DNA very tightly, giving it that pinched look. It's also at the centromeres that we get attachments to the cytoskeleton that will be important, as we'll see, in separating the sister chromatids from each other. It's worth pointing out here that most of the time the DNA is not tightly coiled and tightly bound up into chromosomes that look like this. Instead, the DNA is only loosely wound around proteins and exists as very stringy DNA in the nucleus. It's only as the cell gets ready to divide that the chromosome binds up tightly like this. This lower picture shows how the chromosome varies with time. An unreplicated chromosome is spread out and not condensed. It's a single double helix. When it's replicated, the double helix becomes two double helices, and individual genes along the strand of DNA are also, of course, copied. When the cell enters mitosis, the sister chromatids become condensed and gain that classic X shape as the centromere is particularly tightly bound. Let's look at what mitosis accomplishes in a cell. Perhaps the most important thing to get out of this is the distribution of chromosomes and how the division of the cell ensures that daughter cells are both identical to each other and identical to the original mother cell. Remember that the G1 phase is when the cell is just sort of doing its thing. If it's an islet cell of the pancreas, then it spends its time pumping out insulin. Notice here that during G1, there are two copies of the single chromosome. This is a homologous pair. Notice that they're drawn to be the same size. Each of them is a continuous double helix of DNA. During S phase, each of those chromosomes makes a copy of itself so that pairs of sister chromatids are joined together at the centromere. During mitosis, the sister chromatids are separated in such a way that one always ends up in one of the daughter cells and the other always ends up in the other daughter cell. The end result is two cells that have the exact same genetic makeup as each other and the exact same genetic makeup as the original mother cell. Remember, mitosis is just a cellular duplicating process. Let's start looking at the details of mitosis by starting out with what the cell looks like just before mitosis starts. In a G2 cell, the chromosomes are uncondensed and stringy. They haven't yet formed into the tight structures that we saw earlier. Another important feature of mitosis is the role of the microtubules of the cytoskeleton. Remember when we learned about the cytoskeleton, we noted that there are two centrosomes and each has a pair of centrioles that are oriented at 90 degrees to each other. The centrioles are the organizing center of the microtubules. Remember that microtubules have a plus end and a minus end. The minus end starts at the centrioles and the plus ends go outward from there. The first stage of mitosis is called prophase. And during prophase, the chromosomes become condensed and bound up into discrete chromosomes. The centrosomes also split up and head to opposite sides of the cell. Microtubules extend out from the centrosomes and form what is called the mitotic spindle. During the next phase, prometaphase, the nuclear envelope disintegrates. This allows the microtubules to reach into the nuclear space, and they attach to the kinetochore. The kinetochore is a protein complex attached to the centromere of each sister chromatid, and is the point of attachment for the microtubules. The microtubules begin to tug at the kinetochore of the sister chromatids. The next phase is metaphase. During metaphase, the chromosomes are pulled and tugged by the microtubules of the spindle. This pulling and tugging aligns them in the center of the cell along what's called the metaphase plate. Remember that the cell is three-dimensional, not two-dimensional as we see here. Therefore, the middle region is basically a disc that's called the metaphase plate, along which all of the chromosomes line up. At the beginning of anaphase, molecular signals cause the cohesins, proteins that bind the two sister chromatids together, to loosen. Then the microtubules shorten, and the sister chromatids, now considered to be chromosomes in their own right, separate and travel toward the centrosomes at the poles of the cells. In telophase, the nuclear membrane reforms with all of the chromosomes within it. Subsequently, the chromosomes start to decondense and go back to their stringy appearance. Once the nucleus has reformed, mitosis is complete, and the cell now has two distinct nuclei, and each of these nuclei are exact replicates of the other. The final thing that needs to happen before the cells are completely divided is cytokinesis. During cytokinesis, actin filaments tighten around the cell, causing a cleavage furrow, and that tightening eventually pinches the cells in two. This process is different in animal cells than plant cells because plants have a rigid cell wall and can't be pinched in two. Instead, a new cell wall is simply built between the two new cells. Looking in a little bit more detail at how the microtubules control the movements of chromosomes, two hypotheses have been proposed. In the first hypothesis, the microtubules shorten at the centrosome. When they pull the chromosomes, it's like they're reeling them in like a fisherman reels in a fish. The second hypothesis is that the microtubules shorten at the kinetochore on the chromosome end. With this hypothesis, the chromosomes sort of crawl along the microtubules. A really nice experiment was done where the microtubules were given a fluorescent label to make them glow yellow. Then a laser was used to eliminate the yellow fluorescence from certain regions of the microtubules. As anaphase proceeded, the researchers noted what happened to the dark, non-fluorescent part of the microtubules. You can see that if the first hypothesis is correct, that microtubules shorten from the centrosome at the edge of the cell, then this dark patch on the microtubules would move outward toward the periphery of the cell as the chromosomes are reeled in. If the second hypothesis is true, then the chromosomes would move along the microtubules and eventually pass the dark patch on the microtubules. The results from this experiment showed that the chromosomes walk their way up the microtubules. The dark patch did not move. The proposed model for how this works involves the kinetochore and motor proteins attached to the kinetochore that walk along the microtubule. As the microtubule passes through the parts of the kinetochore, it disassembles into its tubulin subunits. As this happens, the chromosome gets dragged behind the kinetochore. Although this seems to be the primary way that chromosomes move during anaphase, in some cells there does seem to be some reeling in action and shortening of the microtubules at the centrosome end. Perhaps some cells predominantly use one mechanism and others use the other. Before we finish, I just wanted to mention about binary fission in bacteria. Bacteria have one or more circular chromosomes, each with an origin of replication. When the DNA is copied, it's copied from this origin of replication, and as a copying continues, the origins of replication separate to opposite ends of the cell. Bacteria don't have microtubules like eukaryotes do, so this migration of the origin of replication is important for separating the two or more chromosomes. After the chromosomes are in the opposite ends of the cell pinches off and the two daughter cells are independent.